Well, thank you very much for the wonderful introduction. I've been coming to PICON since my second year of graduate school, and I've been coming to these talks. So I can't explain what an honor it is to be up here receiving the PICON Achievement Award. And it's a real privilege to be able to share some of my group's activities with you over the past few years. So I'm going to share a story that I'm really excited about. And here's a story where we've tackled a pretty important public health problem, and we're coming at it from a basic chemistry perspective. So we've developed tools to ask really relevant questions and provide very important information to the field. So that important problem is depression. Depression is a mental health issue that affects a phenomenal amount of people, and it's on the rise globally. It's very difficult to diagnose. There's no chemical test for depression. Despite this, 1 in 5 Americans are prescribed a frontline treatment for depression. That's the selective serotonin reuptake inhibitors, SSRI. The SSRIs have become under the spotlight over the past few years because they have very variable efficacy rates. So they don't work for about 70% of patients that take them. There's a lot of dosage problems. There's a peculiar problem that when patients take them, they have to take them for 2-3 weeks, sometimes longer, before they see a therapeutic efficacy. And because there's no chemical test for depression, there's no chemical test to screen antidepressants. So there are a lot of issues here. And most of these, if you want to ask why these persist, there's a really simple answer to that. And the answer is that we don't really understand the fundamental chemical underpinnings of depression. And there have been different theories that have been around. So this theory, the monoamine theory of depression, has been around since the 70s. And so in a nutshell, the idea is that there's a focus on the molecule serotonin. So in the brain, serotonin acts as a neurotransmitter, more specifically a neuromodulator. And it's responsible for relaying biochemical messages from a presynaptic cell to a postsynaptic cell. So here is a normal cell. The serotonin is released from vesicles into the cell. It relays its biochemical message through interaction with these postsynaptic receptors. The idea is that during depression, for some reason, the serotonin levels in this space, which I'm also going to call the extracellular space, are lower for some reason. And that administering SSRIs, which will block the reuptake of serotonin back into the cell, will raise the serotonin levels back to normal or even higher. So like I said, this theory has been around since the 70s, and there are several issues with it. So I'm going to take you through some of the issues and then talk to you about what we've done to look at it. So the first issue is that despite this hypothesis being around for such a long time, there's really no consensus in the field of serotonin's actions during depression. We don't know yet whether serotonin is lower during depression. I'm going to show you some data that I think will put this issue at rest. But for the time being, there has been no consensus, and that's because it's very, very difficult to measure brain chemistry. The traditional analytical methods have looked at kind of a slower chemistry that's perhaps not relevant to neurotransmission timescales. It's often done at large probes that cause tissue damage, so there's inconsistencies in the data. That's the first issue. The second issue is this idea of antidepressant variability and the delayed onset of action. So the SSRIs are designed to block the serotonin transporter, thereby increasing the extracellular serotonin levels. So if the serotonin hypothesis holds, they should work. They should work for more percentage of patients that take them. And then this idea of the delayed onset of action, like I said, is peculiar. Patients take the antidepressants for several weeks, and in that time, the patients often feel much worse than they did. So if this was some sort of gradual raising of the serotonin levels, it's not consistent with this real-life phenomenon. The third issue is screening antidepressants. So like I said, there's no chemical test. And so it's very difficult to screen antidepressants and have an indication of their clinical efficacy. The way that this has traditionally been done is using behavioral tests in animals. So what I'm showing you here is an example of a forced swim test in mice. The mice are put into a vessel from which they can't escape, and they will swim, try to get out, and at some point after a few minutes they kind of give up trying and they go into a learned helplessness state. If you give these guys an acute injection of SSRI into their interperitoneal cavity, they'll swim for longer next time. That's only a robust behavioral shift in animals that are not depressed. When animals become depressed, this behavioral shift is no longer as robust. So again, there's this real inconsistency with the serotonin hypothesis of depression. The final thing I want to touch on is this idea of translating the information that for the majority has been done in mice. So most of the depression research has been performed in mouse models or rat models. And of course, we're really interested in translating that into human models. And so what I have here is kind of a status quo of what we know about depression in mouse models versus human models. So let me just take you through this real quick. In terms of etiology, that's the cause of depression. Causing depression in mice and in humans is very similar. So mice will become depressed if they experience stresses, chronic illnesses, there's a genetics component. These are very similar in humans. That's where the similarities end. In terms of diagnosis, in a mouse there are a well-established battery of tests. I showed you one of them, the forced swim test. In humans, the diagnosis is, for the most part, questionnaire-based. A healthcare practitioner will ask questions and then provide a diagnosis. And then in terms of treatment, there's a real disconnect. So this acute interperitoneal injection in naive animals causes a behavioral shift in mice. If those mice are now depressed, there's much more variation in their behavioral response. And it's very, very difficult to give mice a chronic administration. It's almost impossible to mimic the human administration. This is the once daily tablet. The mice won't take it. So in humans, we have this chronic SSRI administration, the low dose daily tablet, with very variable effects. There's focus in recent years on ketamine, and this is an IV one-time dose of ketamine that has some variability in its efficacy. I'll come back to that. And there are other treatments. I put another one here, electroconvulsive therapy. So you'll see there is this disconnect, and we really have ideas about how to translate the mouse work into the human work. So I'll talk to you guys about that too. Okay, so this brings me to kind of an outline of the things that I want to tackle, and these are no small things. These are big questions, and we've been working on this for a long, long time, and I think we're finally getting to the point where we have some good answers. So we want to start thinking about how to really sort of robustly diagnose depression. And to do that, we need a testable hypothesis, and we need a way to test that hypothesis. So here's where the analytical chemistry would come in. We want to really look at this idea of the treatment, and for that we want to look at this sort of time frame issue, so why it would take several weeks, can we speed that up, where does the variability come in, and then we want to start thinking about high throughput, robust in vitro methods for screening antidepressants that is actually a chemical test. And in all of these you'll see I put this idea of translatable, and we'll come back to that. Okay, so when our approach to this is to look at the chemistry, like I said, the fundamental chemistry of the brain. And so this is a very nice image that my student made for us, but essentially she's showing the technique that we use. This is the carbon fiber microelectrode, and we couple it with a couple of different techniques. And we're interested in looking at serotonin chemistry because we want to sort of focus on that monoamine hypothesis. So over the past few years, we've created some very nice, very unique tools to look at serotonin chemistry. There's a couple of things when I think about neurotransmitter chemistry that we want to look at. So there are sort of ambient levels of serotonin that don't change very rapidly. They're there in the extracellular space, but there are also bursts of activity that come out of the neurons. And so that's kind of the two types of chemistry are the slow chemistry that we want to look at and the faster and more evoked type phasic responses that we want to look at. And so our technologies really, like I said, they're very unique, they're very, very robust, and they've given us some really important information. So I'm going to talk to you about measuring fast chemistry with a technique called fast-scan cyclic voltammetry, and the slow chemistry with a technique called fast-scan adsorption controlled voltammetry. Okay. So jumping into fast-scan cyclic voltammetry, slides like this you've probably seen people who do FSCAV present. I'll just take you through this real quick for those of you not familiar with the method. This is a carbon fiber microelectrode. We hand fabricate these, and we will manually insert that directly into the brain tissue of intact, sleeping, or freely moving animals. We will apply a waveform to this, and these waveforms can differ based on the analyte that you're interested in. A lot of work was put into kind of creating this waveform that is very selective for serotonin and reduces electrode fouling and has other advantages. This is applied to the electrode, and it will capture the 2-electron oxidation reduction of serotonin. The electrochemists will notice this is a very, very fast scan rate. And with a scan rate like this, we can capture this process within a couple of milliseconds, and it'll give us this cyclic voltammegram. Like I said, it takes a couple of milliseconds to capture this cyclic voltammegram, and so we can present the cyclic voltammegram as current versus time. That's the 2 milliseconds it took to collect. And then we'll stack these next to each other now. We wait 100 milliseconds between each scan to allow pre-concentration processes on the electrode surface. And then we give this guys a false color, so anything in green coming up out of the page is an oxidation, anything in blue going into the page is a reduction. And we kind of look at this top down from like a bird's eye view. Here there's an event. If we didn't know what that event was, we could take a vertical stripe through it, reconstitute the cyclic voltammogram and know what we were looking at, selectivity. And then more physiologically relevant would be a horizontal stripe through the peak oxidation current that would give us the concentration versus time. Okay. FSCAV is background subtracted. When you scan so fast, 1,000 volts a second, if we were to take a measurement, all we would measure is a charging current on the electrode surface. So we have to background subtract all of the cyclic voltammograms to see the Faradaic processes, see these under there. And so that means to get meaningful data, we have to evoke a change. And so this is a representation of the in vivo model that we'll use. So this is a mouse's brain. His eyes would be here, his tails would be here, his tail would be here. And so we'll stimulate an axonal bundle that comes out of a cell body region. So this is an area where serotonin is made. The axons traverse through this area, the MFB, and then they terminate in terminal regions that we're interested in. So today, primarily, I'm going to talk to you about work that we've done in the hippocampus and in the hypothalamus, two very important regions for depression research. Okay, so real quick, I don't want to spend too much time on this slide, but there's a one particular take-home message I want to give you guys before we move on. So this is what in vivo data looks like. This is a evoked serotonin response in the hippocampus. Here's the cyclic voltammogram. And here is data showing the responses in male and female mice, averaged. And so you see the serotonin increases during the stimulation, electrical stimulation, and after the stimulation, the serotonin is cleared. And so we try to model this response. And the primary clearance mechanism is that serotonin transporter that I showed you guys in the first few slides. And that's the protein that's targeted with the SSRIs. But it turns out we couldn't purely model it just using that one reuptake mechanism. We had to have two reuptake mechanisms. So we have two Michaelis-Menten curves here in the model that describe this curve. And what we discovered, and it's been sort of postulated before and verified thereafter, is that actually other monoamine transporters, including the dopamine transporter, the norepinephrine transporter, and an emerging class of transporters called the organic cation transporters, will also clear serotonin. So what I want you to take away from this slide is that monoamine transporters are promiscuous. They'll reuptake each other's analytes. That becomes important as we go on in the talk. All right, so real quick, I want to talk to you about this technique that measures the ambient levels of serotonin. So like I said, FSCAV is background subtracted to get rid of this large charging current, which means we have to evoke a change. We don't know where we started. So this is some work that I did with Michael Hine at the University of Arizona. And this is my graduate student, Aya, who's now working for Babic at Brighton. She graduated. And so we developed this method where this is work that we published for dopamine, but we recently published our serotonin work. And so essentially you take the same carbon fiber. You don't have to change anything. You just make a small modification to the detection scheme. If you apply the waveform now at a very high frequency, you essentially can minimize the adsorption to the electrode. So we assume there's not much happening at the electrode. Then you turn the waveform off for 5-10 minutes and allow adsorption to reach equilibrium with its surroundings. And then we'll apply the waveform back, and now that's that specific waveform for serotonin. So everything that's on the electrode surface will become oxidized, and the first few cyclic voltammograms will allow us to quantify what's on the surface, which is the ambient levels. All right. So there are the techniques. Now I want to get to the really good stuff. And so, sorry, this is just one slide showing you how Aya has quantified the ambient levels of serotonin in the hippocampus at about 60 nanomoles. And this is a comparison of the in vivo versus in vitro responses. Okay, so I want to show you this slide, and this is the culmination of many, many, many years' work. And it doesn't look like a very fancy slide, but it is, okay? And so this is the big question, and it's really, I can't stress the importance of this question. People have been trying to answer this for a long time. So here's the question. Is serotonin actually lower during depression? And of course, we have mouse models for this. We can't test this in humans yet. Mice become stressed. There's different ways to stress out mice and get them to have depression phenotypes. One very robust behavioral model is called the chronic unpredictable mild stress. Here, mice are exposed to 3 unpredictable stressors daily for several weeks to several months. And after some time, they start to show depression phenotypes. So we measured their ambient serotonin levels. Here are normal mice. So here's a happy mini. I have to. We're in Orlando, right? So here's a happy mini. There's a spread of data that I also showed you on the previous slide. That's sort of ambient serotonin levels. And my student Melinda collected this data where she shows, now we haven't quite gotten enough to do a lot of statistics on it, this is brand new data, but indeed in the chronic mild stress guys that have depression phenotypes, their serotonin is lower. Okay? So this is a huge finding, like I said, many, many years of work. And so really this begs the question, is the same true in humans? And I'll get back to this, okay? But this is kind of a big deal so far. First question answered. Let's get to the second question, and this is more complex, okay? So at this point, we're kind of now fans of this monoamine hypothesis. We're like, yes, we think it's right. So now let's get on to the treatment problems. So we want to start thinking about SSRI treatment, and let's tackle this idea of the time delay. Why do people have to take it for 2-3 weeks to see a therapeutic effect, okay? I want to start with data from Whiteman's lab from the 1990s. And here they would take the brain out of a mouse, they would slice up the brain, keep it alive, and go to an area of interest. Here they're looking at 2 areas where serotonin is present. They stimulate serotonin release, they perfuse an SSRI over that preparation, and then they see a very sensible reuptake inhibition. The slope is slower, okay? Very sensible. We tried to do this in vivo, and we got decidedly asensible results. Well, these are very robust. This is what happens, but we're having a hard time understanding it. At least we did have a hard time understanding it. So let me take you through this. What we've created here is a dose-response comparison. So a dose-response, for those of you not familiar with pharmacology, you have a drug, you want to know, you take an index of the drug's efficacy. Sometimes it's behavioral, sometimes it's chemical. And then you'll increase that dose and plot that index of the efficacy versus the increasing dose. Okay, so this is kind of what we're trying to do here. We give a relatively low dose, 1 milligram per kilogram, and we notice something relatively instantly. So this is averages of 5 mice. We notice that although there's uptake inhibition, it's relatively simple to explain the response. There's no steady state response that we can take, so the response changes. After 5 minutes, it's different than 10 minutes, then 20 minutes, then an hour, then 2 hours. So it became difficult for us to decide from the outset at which time point to take that index. And actually, we didn't know which index to take. Do we take the reuptake curve, or do we take the increase in amplitude? But the situation got infinitely more complex when we gave a higher dose. So now this is 3 milligrams per kilogram. It's an intermediate dose. It's by no means a high dose. And you can immediately see there are issues here. The very first response of the brain to this intermediate dose, the 5-minute response, is actually an increase in the rate of reuptake. Well, this is crazy because the drug should decrease the rate of reuptake. It's sitting in those serotonin transporters, right? 5 mice, this is robust data. The response slows down over 120 minutes, but all of these curves are slower than they were for the 1 milligram per kilogram scenario. Increasing the dose, again, has this funny phenomenon that we don't reach a steady state, and at the 30 milligrams per kilogram dose, you're getting to be a high dose here. You see it after 120 minutes, there's almost no reuptake. So now this is not reuptake inhibition, this is no reuptake, all right? So we were stuck with this for a while. And looking at this, for me, this told me these rapid changes in the reuptake rate. This told me that there are things happening to the serotonin transporters on the cell surface. Okay, this is what this told me. So I made some hypotheses, and I found a group in Germany. So this is my postdoc Rachel, who has been living out in Germany for some time doing these experiments. And so I found this group, and they have a pretty neat preparation. So I'll take you through it. They have stem cell preparations. And they have these cultures under a confocal microscope. They add an antibody that will target the extracellular loop of the SERT, and that antibody is fluorescently active under a confocal microscope. So they look at their stem cell preparations. If there's a decrease in fluorescent signal, it means there's less of those serotonin transporters. If there's an increase, it means there's more. If there's no change, it means that there's no change. This paper of theirs is the one that I found. So basically what they did is they took their cultures, they perfused them with SSRI, and they found over 4 hours there was a decrease in fluorescent signal, which means there was less of these serotonin transporters. This I liked because it kind of mirrors the higher dose signal that I showed you where there was no reuptake. And so they hypothesized that there was a desensitization or internalization of the serotonin transporter over time in response to this acute dose, okay? So I thought that was kind of cool, but I wanted to know, let's do this again, and is there now closer, after a minute or two, is there perhaps an overexpression of these serotonin transporters, right? And they kind of thought I was nuts, but they did the experiment, so Rachel collected this data, and that's exactly what happens. So you have to stimulate the cells, but what I'm showing you here is that in the presence of an SSRI and stimulation 2 minutes after SSRI administration, there's an overexpression of the serotonin transporters. And we collected data in several stem cell preparations, and indeed you can see that when there's stimulation, so serotonin has to be released, and when there's SSRI on board, there are more serotonin transporters. This is really significant, because I believe that this is what's happening during that low-dose chronic regime in humans. I think the brain's first reaction to its increasing extracellular serotonin levels is to express more serotonin transporters on the surface. In this low-dose chronic regime, the brain has more opportunity to deal with that, if you like, and after 3 weeks, they become desensitized. But in an acute scenario, this scenario that works in the naive animals where they get the acute injection, this happens quickly, okay? Another reason I like this is going back to the ketamine issue. So the ketamine treatment is one acute dose in clinical conditions. So could it be that that's what's needed for SSRI therapy? So just a quick in vivo experiment to kind of back that hypothesis up. So we're taking basal levels now in several mice. Here we just take basal levels for an hour. Here we give an SSRI. And you see there's like a 10-minute region where there's no change in the extracellular serotonin levels. But we did see after 5 minutes, we had that increase in the rate of serotonin reuptake. So this is the brain trying to regulate its serotonin levels. Right around 10-15 minutes, it kind of gives up the fight because this is acute and large. And the serotonin levels start to go up because the certs start to get desensitized. Okay, so that speaks to the perhaps the time frame of treatment. Okay, and I think we have some very important ideas there. But it doesn't explain, so I showed you in the depressed mice there's less serotonin, and I just showed you that an acute injection in naive animals will raise the serotonin levels. So it doesn't really explain the disconnect between the variability and the SSRI treatment. But of course, these were not depressed animals. So I'm going to talk to you about variability. I'm going to now jump into neuroinflammation. This is related, I promise, okay? This is a related phenomenon. The new hot topic in depression research is neuroinflammation. And it turns out that there's no depression without neuroinflammation, and there's no neuroinflammation without depression. It's kind of very much a chicken and egg story in the field, okay? This is a very complicated slide, but it's just showing you kind of the chemical underpinnings of neuroinflammation. Neuroinflammation, if you like, is the brain's immune reaction to something. This slide is showing you that it could be a brain immune reaction, but it's also very much linked to a peripheral immune reaction. So people that experience peripheral immune reactions will also experience brain neuroinflammation, which is very much linked to psychiatric disorders, including depression. This is what we kind of know. There's a lot of work on cytokine analysis and stuff like that. All right. So I got to thinking chemistry, right? And there's stuff we know about peripheral inflammation. It's much easier to define peripheral inflammation. What we do know is histamine is the signaling molecule of peripheral inflammation. So this is a simple schematic just showing you that histamine is released, and it sets into motion several events that can deal with that inflammation. What is not known is the role of histamine during neuroinflammation, because it's not very easy to measure histamine. So the work that I'm going to show you now was performed by my graduate student Srimal, and who's now a postdoc at Vanderbilt, and my third-year graduate student Melinda, who pulled off a heroic feat this week by getting this data. So Srimal, while he was with me, developed a really cool method using FSCAV to simultaneously measure serotonin and histamine. And so he went to a hypothalamic region where both were present. He stimulated histamine but not serotonin release, and he found that histamine goes up, gets cleared, and in response to it there's a decrease in the serotonin levels. We did a whole bunch of modeling and pharmacology, and we found that this process was mediated by inhibitory H3 heteroreceptors on serotonin terminals. So the histamine is released and acts to inhibit the serotonin firing, okay? So now bringing stuff together, here was my question. We know histamine is increased during peripheral inflammation. If histamine is increased during neuroinflammation and histamine is inhibiting serotonin, this could have real implications. So we really quickly answered this question. We had 2 models of inflammation available to us, and we've expanded this now, but this was the very first thing that we did. We have some HIV transgenic rats that we're doing work on for another project. We took 7 of these aged rats that have a low-grade chronic inflammation. We measured their histamine and serotonin profile. The blue is in the control, the red is the inflammation state, and you can clearly see there's more histamine, and as a consequence, there's more serotonin inhibition. This is really cool, this acute neuroinflammation. Here, we peripherally injected LPS. LPS is like a polysaccharide. It is the molecule found in the outer membrane of bacteria. When you inject it peripherally, there's a violent acute immune reaction. It does not go through the blood-brain barrier. We inject it peripherally. 5 minutes after peripheral injection, we see an effect in the brain. So the histamine levels in the brain are increased. As a consequence, there's more inhibition of serotonin. Okay, so we think there's more histamine during inflammation, and we think it's driving down the serotonin. But that doesn't explain the variability yet, but I think I'm going to start to get to that in a second. So here is Melinda's beautiful data that I'm going to expand on. So Melinda did basals in several mice, basal levels. These mice here in blue are not experiencing inflammation. So at this point she gives these mice saline, nothing much happens. At this point she gives them the SSRI, and as we saw before, there's an increase in the serotonin levels. In a separate group of mice, she gives, instead of saline, LPS, that I showed you would increase the histamine levels. The basal levels of serotonin drop. And when you give these guys the SSRI, they don't go up as much as in a non-inflammation state, right? That's really cool. So we're showing in inflammation states, SSRIs are not as effective in raising the serotonin levels. Here's a chronic model of inflammation. This is now our stressed out guys, the chronic mild stress guys that I showed you had less serotonin. So they start off with less serotonin. We give them saline, nothing much happens. We give them escitalopram, the serotonin levels go up, of course, but they don't go up as much as the naive guys. And then if you merge these, it's kind of cool. The 2 inflammation states have the same low level of serotonin. And again, giving SSRI doesn't allow the serotonin levels to come back up to normal. Why? I think we know why. So histamine is a relatively understudied neurotransmitter. And if you go through the literature, people have not identified a transporter for histamine reuptake back into the cell. There must be, because histamine degradation is exclusively intracellular via enzymes, okay? So I got to thinking, let's go back to this idea of the promiscuous monoamine transporters. Something is reuptaking histamine. So we inhibited everything we could think of. We started off with a serotonin transporter, and these are now histamine traces before and after administration of the drug. And we saw an inhibition in the histamine reuptake, so we were like, yeah, it's serotonin transporter. I'm glad that we didn't stop there, because we used different inhibitors of the serotonin transporter, and we saw that effect to varying degrees. We were less sure that it was a serotonin transporter. We gave the dopamine transporter inhibitor, didn't see much effect. We gave a norepinephrine transporter inhibitor, we saw some effect. And we gave the organic cation transporter inhibitors, and we saw an effect. I'm going to tell you that we think it's the OCTs. We're not there yet. We're using some genetic models to verify that. But what I can tell you is that these 4 compounds that inhibit histamine reuptake have antidepressant activity. That's phenomenally important. Because if we are saying that serotonin levels are lower during depression, and the frontline treatment to increase the serotonin levels is also increasing histamine levels, which is inhibiting the serotonin levels, you can clearly start to see how this speaks to the variability in the response. Okay? So the final experiments done by Melinda the hero 2 days ago, or 3 days ago, to really show that are these. So here are these guys that were the control guys. We give them saline, nothing much happens. We give them the SSRI, the serotonin goes up. The LPS guys, the serotonin went down and then didn't go up very much after the SSRI. If here, in conjunction with the SSRI, we give an agent that inhibits histamine synthesis, we get the levels going right back up to the naive guys. And for the chronic scenario, the CMS guys, the behaviorally depressed mice, if we give histamine synthesis inhibitors along with the SSRI acute dose, we get the serotonin levels going right back up to where they were. So we have managed to kind of get the serotonin levels back to normal by inhibiting histamine synthesis. Okay, almost done. We've answered some important questions. We think we can diagnose depression by measuring serotonin levels. This is kind of cool because people have been thinking this for a while, but we can do it now. we think we have some ideas about how to improve current treatments. And so SSRIs are kind of going out of fashion for researchers, but I think by having this histamine story, we can really reinvigorate the field. The time frame idea is the overexpression and underexpression. We can probably speed that up with acute doses. The variability takes into account the histamine. I've got some question marks up here for the translatable aspects of this. So everything I showed you is in mice. We can't make measurements in human brains. That's not going to happen. So we have some ideas on how we can translate our findings. This is the final slide. I was in Germany last week with Rachel, where we're collecting this data, and I'm super excited about it. So our collaborators in Germany, they're the Mannheim Institute of Mental Health. They have a very unique setup. So they can differentiate stem cells into serotonin neurons. That's very unique. More than that, they can take human stem cells and differentiate them into serotonin neurons. And so they're very cool. They create networks, and they're already sort of starting to create micro structures of the brain too. So these are really unique setups, if you like. Here's the setup that Rachel and I worked on. So this is a photo we took with a cell phone, so it's not very beautiful, but it illustrates what I want to show you guys. So these are human stem cells. They've been differentiated into serotonin neurons. And they kind of create these clumps of cells. They're both differentiated and undifferentiated cells here. And you can see they start to send projections to other clumps. So they're creating networks, they're creating circuits, okay? I wanted to use this setup. And here's a stimulating electrode that'll go around this clump. And here's a working electrode that'll be in this clump. My question was, can we measure serotonin out of these cells? If we can, does SSRI administration look like it does in vivo? Remember, when we take the brain out and slice it up, we don't get that SSRI response that we do in vivo, okay? And I think the answer is yes. Now, this data is not very beautiful. We're very, very, very tiny signals, but we can measure serotonin. And so here we have averages of 5 stem cell preparations. So what I'm showing you is the control response here. We infuse SSRI over these guys at 0.1 micromolar, and this is an average of 2 files that were taken 5 to 10 minutes after, so that rapid response. You see that the lower dose sort of does that uptake inhibition like we saw with the lower dose in our in vivo model. But the higher dose increases the rate of reuptake. So this is the higher dose here, and that's what we saw in the brain, right? And then as you increase the dose, we start to get a lowering in response. So this is really, really exciting for me. Going back to our issues and prospects, we now have a method that is in vitro that is high throughput. It truly is translatable because we can take patients' own cells. We can do 2 things with this. We can then make these diagnoses of depression, and we can screen in a high throughput method new drugs based on our own monoamine hypothesis of depression, take into account the histamine, and also we can know per patient, in terms of personalized treatment, what would be the best strategy for treatment. Okay, so that's the end of the story. Like I said, my group pulled off a heroic effort. They're superstars to get this data together. We've been working on this for many years. A lot of the stories came together in the last couple of weeks. The data that I showed you was primarily collected by my postdoc Rachel, who came to me from Sulanti's lab, Aya, who's now at Babik's lab, Melinda, and our collaborators on this particular project, and then our funding sources. So I'd like to thank you guys again for your attention, and thank the committee for the award.